That can't happen when you're a diabetic. The last part, you want to stay away from the fuels that make less water. Everybody knows carbohydrates only, if you take it on a molar level, they only make 55 grams of water, where TCA cycle makes about 105. So remember, this story is about reestablishing water production at CCO. So of course we need more water. This is the reason why, what do diabetics get, Nick? You've heard the story when you were a young man: polydipsia, polyuria, polyphagia, because the mitochondria are broken. They eat more because they can't make the water and they can't get rid of the bad engines. They piss more because they can't retain their stuff at the hypothalamic level. Why? Because vasopressin is broken for them. Because what controls that? The central retinal pathways that we talked about earlier. In other words, everything is shot. So they are like looking at a plant in your backyard that has 19 holes in it, and your mom and your girlfriend keep putting the water and the nutrients in, and it just leaks right through. Mm-hmm. That's what they are. And all we are, when you understand the story I'm giving you, we are a little bit more complicated than a houseplant. That's what hemoglobin and melanin really gave us from chlorophyll. And when you understand that the same things that control regeneration in plants control regeneration in us, it's the same story. DC electric current, Becker showed, was present in plants. So did Burr, where Becker got all these ideas. And then when Becker started to find out that hemoglobin was tied to this, the first thing I did when I read his book, The Electric Body, in 85, I looked at that picture that you mentioned earlier. Man, this porphyrin ring looks really similar. You know, there must be a story here. And the story was, I thought the story was always going to be about the different number of electrons and the photoelectric effect. Turned out that's part of the story, but the real part is the paramagnetic switch. That's the real big part of the story.
So when the animals, the asteroid hits, the dinosaurs die off, and the subterranean animals start to come up, they enter into what is a new and now changing oxygen and light environment. Walk us through the radiation of mammals from your perspective.
Yeah, what happens then is the mammals now have a normoxic environment. They're no longer hypoxic as they were. The melanin that was outside in their fur and their hair actually begins to get sucked inside. Why is that the case? They're now sensing UV light. What's the UV light sensor that innovates probably at the time of the Cambrian explosion? NeurOps, I don't know how much you know about that.
Very little.
But it's a UVA detector that's present on mammals' corneas and their skin. And as you lose hair, what happens? You become better UV light detector. Mm-hmm. So this is the stimulus for POMC. What is POMC? Pro-opiomelanocortin. It's a gene in mammals that is really what we specialize in. And it turns out what translates POMC from our DNA? UV light's the stimulus. Where does that light come from? Most people think that it's from the sun. I'm going to tell you, in the original animals that first come out, it's not only from the sun, but it's also from the ultra weak biophotons that they make, which tends to be in the ultra weak spectrum, which is 200 to 400. How do we know that? Because everything that is about a mammal, which is the leptin melanocortin pathway, and then this hemoglobin story with the intermitochondrial membrane becomes sculpted. That's where you have NAD to oxygen. What's the change there? Negative 400 to positive 400 volts. But what's the other thing? What's the emission and absorption spectra of cytochrome 1? Turns out it absorbs at 340 and it emits in the blue range, in the flavin range. That's why the second cytochrome is a blue absorber. It also turns out that it makes the most superoxide pulse in all of the mitochondria, which, what are free radicals? Remember, they're unpaired electrons. What does that mean? It means they all have absorption and emission spectra. So the other problem in biochemistry we haven't got to, but I did sit down with a famous biochemist probably 10 or 15 years ago that you probably have heard of, Ray Peat. And I tried to tell him this story, and his eyes were glazed over. When he told me all the things he said, I said, Ray, I said, do you understand that anything that has an absorption and emission spectrum means that light has to be part of the story? And I told him how old the story went. I said, do you know who came up with this idea originally? Said it was a guy who also won a Nobel Prize for being wrong about the Krebs cycle, which was Albert St. Georgi. And he said the most interesting thing at a meeting in 1941 in Budapest. He said, the only thing that DNA codes for is proteins. And he goes, it's really funny when you look at a protein, they have an electronic structure that mimics a semiconductor. Do you know who was sitting in the audience that day? Robert O. Becker. Literally within less than 2 decades, he proves that Albert St. Georgi is correct. Okay. And but beyond a shadow of a doubt. So when I gave the story to Ray, I said, Ray, all the things that you believe are based on this paradigm of you learning about all these boxcars. But as you said, Nick, in the beginning of this podcast, you're like, Jack, it's not clear to me how the two domains of life came together, which is now you're asking me the next question, which is beautiful, because I know you're understanding what I'm saying now. So let's talk about what happened to these mammals. Turned out these mammals went from being hypoxic to TCA masters with oxygen. And what did that mean at the mitochondrial level? That means they made chemicals and selected for chemicals at a quantum biologic level, not at Darwin's level, that had emission spectras that were all in the UV range.
And now if the switch happens, you now have the energetic potential to build more and bigger complexity.
You got it. And that's exactly what the story of E equals MC squared is. You know, I don't have to teach you about that because, you know, energy and mass and Einstein's equation are the same. So the thing is, if you're able to have more energy in the system, which is what Pierre-Jean's theorem is, he won the Nobel Prize in 1977. Basically, a cell is a quantum cell that's designed to pump light into it. And what do we do? Through electrical resistance, photoelectrical resistance, that's how you build the complexity. So what are you doing? The electrical resistance of things in a cell slows the light down. Those things harvest the power, and then we build the complexity from that. It's no different than the story I told you about what happens to a baby when it's inside its mother. Okay, it's exact same story. It's another fractal of the story. And when you see it for yourself, you start to look at biochemistry. You start opening up your Leninger biochemistry and go, Jesus Christ, I never learned anything about absorption and emission spectras. And this is the reason why, Nick, I'm such a pain in the ass for guys like you that are in evolutionary biology and biochemistry, because I keep pointing out these key features that you're forgetting. And you guys know a lot of the stuff I know. You know about the GOE. You know about the Cambrian explosion. You know about Darwin and how the story doesn't marry up to the Cambrian explosion. But because it's central dogma, nobody wants to question it. Why? Because if you're a PhD, you'll never get any money from the NIH or from DARPA or for anybody else. And I understand that. But remember, we're doing this podcast not to talk about how centralized science went off the rails. We're actually trying to teach people the story of life. Yeah, that's what the story about, and how we're able to regenerate, how we're able to wound heal, how we lost the ability to regenerate as we became obligate TCA animals that used oxygen. Doesn't mean we've lost the ability not to do that. How can I tell you that's true? When a kid comes in and drowns, like say where you live, in cold water, we code them all the way for 45 minutes because we know the other free radical chemicals can act as terminal electron acceptors, like hydrogen sulfide, carbon monoxide. We can still save those kids, so we don't give up on them. But the thing is, that's from our evolutionary past. We lose that ability as soon as they cut the umbilical cord and the kid takes its first breath of air.
Because then the hypoxia is gone. This metabolic switch happens, and it just switches everything.
You got it.
And this is why you use the word
environment. Yeah. You change your environment to what we've done now, you begin to realize that the electromagnetic signals outside are doing things to that oxygen-oxidation state on the inside. And what do we know from Nora Volkow's work? We know that non-native EMF and blue light, what's the fundamental thing they do? They cause a drop in NAD positive and pseudohypoxia. That's all you need to know, because what happens then? Blood glucose spikes, so does insulin, and guess what? Then all the food gurus are like, oh, it's all about food. No, it's not. It's all about light, and it's always been about light. But you know what the problem is? All the food gurus learned from who? The biochemists. That's actually, if you want to know the most interesting part of this story for me that recently happened, a guy named Cameron Borg, who runs his own podcast, interviewed Andrew Marino, who, Marino was Becker's right-hand man. And the thing that I was really disappointed with in that podcast is Marino clearly didn't do any of Becker's work. All the stuff that I just told you, I basically finished his thesis. But here's the really sad thing.